Hi, my name is Madison Rogers, and I'm in the Fantuzo lab at the University of Colorado Anschutz Medical Campus. Platelet-derived growth factor receptors, or PDGFRs, are a family of receptor tyrosine kinases, or RTKs. The PDGF receptor family consists of two receptors, alpha and beta. Given the structural similarities of these receptors and the fact that they interact with a similar subset of signaling molecules, a critical question in the field is how biological specificity is defined to generate unique cellular responses downstream of PDGF receptor dimer-specific engagement. Here we utilize a novel bimolecular fluorescence complementation, or BIFC approach that overcomes previous technical limitations and allows us to both visualize and purify individual PDGF receptor dimers. We clone plasmids expressing C-terminal effusions of individual PDGF receptors with BIFC fragments corresponding to the N-terminal or the C-terminal regions of the venous fluorescent protein. The individual N and C-terminal fragments are non-fluorescent when bound to monomeric receptors. However, upon ligand binding and dimerization, the N and C-terminal fragments co-localize, resulting in a functional venous fluorescent protein. This complementation event can then be visualized or purified. Using this approach, I generated two stable cell lines to integrate PDGF receptor BIFC sequences into the genome. I did one for alpha homodimers and one for beta homodimers. And I did this using HCC15 lung cancer cells, as they do not endogenously express the receptors and they have little to no PDGF ligand expression. So we first wanted to confirm this complementation event through fluorescence microscopy. We found that upon 5 minutes of ligand treatment, we had venous signal in both of the cell lines and then no venous signal in the absence of ligand treatment. Next, we wanted to confirm the complementation event biochemically. To do this, I performed an immunoprecipitation with a GFP trap nanobody, which will only pull down dimerized receptors. From this data, we found that the beta receptors appear to dimerize more quickly than the alpha receptors. I then wanted to assess receptor activation within these cell lines. To do this, I performed a time course of ligand simulation and then Western blotted for a specific phosphorylated tyrosine residue within each receptor. From these data, we can conclude that there's little to no activation of the receptors in the absence of ligand, and that both homodimers are robustly autophosphorylated upon addition of exogenous ligand, with the beta homodimers exhibiting a much more robust response at this specific tyrosine residue. We next wanted to assess the endosomal trafficking of the receptors, as we initially observed differences in localization for the venous signal between the homodimers upon 5 minutes of ligand treatment. So here I performed endosomal colocalization studies with RAB5, an early endosome marker, at 2, 5, and 10 minutes of ligand treatment. We found that the alpha homodimers colocalize significantly more at this marker, indicating that the alpha homodimers are trafficked more quickly. We next assessed receptor degradation as a potential route that the receptors take upon trafficking in the cell. To do this, I performed a pre-treatment with cyclohexamide, which inhibits translation in both cell lines, and then a time course of ligand simulation, followed by Western blotting for receptor levels. By 4 hours, we saw a similar extent of degradation for both of the receptors at about 80%. However, the alpha homodimers were degraded much more quickly. Another potential trafficking route that the receptors can take is recycling back to the membrane. So here I investigated colocalization with RAB11, a slow recycling endosome marker, at 60 and 90 minutes of ligand treatment. We found that the beta homodimers colocalize significantly more with this marker, indicating that the beta homodimers are more likely to be recycled. Next, I wanted to assess the intracellular signaling downstream of these receptors in our cell lines. To do this, I performed a time course of ligand simulation, and then this time Western blotted for the phosphorylation of specific effector molecules downstream of PDGF receptor activation, phospho-ERK1/2 and phospho-AKT. For ERK, we saw phosphorylation peaks that remain relatively stable throughout the time course, which is similar to what we observed in the alpha homodimer-initiated phosphorylation of AKT. However, the beta homodimer-initiated phosphorylation of AKT was much more transient. Additionally, in both cases, we found that the beta homodimer phosphorylation amplitudes were much higher than that of the alpha homodimers. So to bring this all together, we wanted to determine how the endosomal trafficking and this cell signaling affected cellular activity. First, I assessed cell proliferation by performing a soft agar anchorage independent growth assay and assessing colony count and colony area. We found that the cells expressing PDGF receptor beta proliferated more than those expressing alpha. I then performed a transwell migration assay, and we found that while ligand addition results in an increase in migration for both cell lines, it had a much more significant effect on the beta homodimer cell line. So to conclude from what I've shown you, we found that the beta receptors homodimerize more quickly than the alpha receptors. And we demonstrated that the alpha homodimers are trafficked more quickly and degraded faster, while beta homodimers are more likely to be recycled. And then finally, we showed that the beta homodimer activation leads to increased downstream signaling and cellular activity. Thank you for listening, and I'm happy to answer any questions.